So previously I made a video about this with Spark for Autism, but now there's a 10K Autism project headed by Simon Van Cohen among others that is really awful, and this guy has a long history assaulting with his false ideas, assaulting manliness and maleness to false things, and how he describes it with autism proving wrong again and again, but still went on. He has said that autistic people have zero empathy and proven wrong and are more primitive minded and a bunch of things. But he's the person put ahead, and he put no one ahead with anyone with actual autism. You know, they do have some people on the boards with it, but not in the head positions. But everyone's talking about how this is wrong from a discrimination standpoint, but I want to talk about how the actual science is wrong and messed up. So in this first part, I'm going to talk about genetics and autism and problems with that in this study. And I'm going to try to really explain it in simple terms, so if you have no background in genetics, you can understand it. So the simplest inheritance is what they normally teach in school, and it's way of an oversimplification, is Mendelian inheritance. And that is, you get 2 copies of every gene. And if they're dominant, we make both of them capital. If they're not dominant, recessive, we make them lowercase. And then we sew them, so we might have RR, or both capitals, meaning they're both dominant, or 2 lowercase ones, meaning they're both recessive, or a big capital letter, meaning that's the dominant, and that one's recessive. And each gene, they say, gives you 1 trait. We're going to go over how all this is this oversimplification later, but in the case of RR, let's say that's the red gene. If you have a dominant one, you have a red flower. If you have a lowercase one, you have a white flower. Well, in the case with dominant genes, when you both are the same type, you have the recessive or you have the dominant trait. But if you have a mixture, one dominant and one non-dominant, you have the dominant trait. That is a way of a simplification. Even Mendelian genes that he studied with peas and stuff turned out not to be fully the case. It turned out to be the first case I have here, which is usually a gene isn't fully dominant, another gene. So instead of having, when you have a recessive and dominant gene, you have something called partial dominance, and that means that it's not fully red. Now they have a recessive gene, it's pinkish, or something in between. Other ones, and this actually occurs with colors of a lot of animal patterns you see with like cows having spots, also stripes and spots on cats and more, is that sometimes one gene randomly turns on and off, and it's just completely random which one's on and off. And sometimes it can be one is a little more biased to one or more less than. But then if you had a red and white gene for flower, half the flowers might be red, half them might be white, or it could be even each cell changes back and forth randomly. So randomly one cell's red, one cell's white, and that can give you a pink color, or it can be little splotches. This random turning on and off can also lead to different skin color patterns on people and birthmarks and stuff like that. So it happens all the time. And then you have the case where the dominant gene is the only one expressed, but it's expressed in the weaker one. So in that case of the red flower, it might be a lighter shade of red. It's not pink, it's not white, but it's still a lighter shade of red. Or there's less red dye, so it looks to you the same, but the red more easily gets leaked out after a while, or decays, or damaged. These are all more common than the simple Mendelian inheritance, actually. And then the other thing is it might be on a sex chromosome. So males usually have one X and Y and one Y chromosome. This is for mammals. For other creatures, there's a different system, and doesn't even always count for all human mammals or other ones. Females usually have an XX chromosome. So if there's a gene on the X chromosome, males would usually only have one of those genes, not 2 of them, and females will have 2 of them, but if there's a gene on the Y chromosome, males usually only ones have it. So here's an example of an image showing this, and we have this red X, and the female in this case has a red gene symbolized by red, and a gene symbolized by white. Half of the male ones just get one copy gene, so you get totally red or white, one genes. But the male has this white gene, so 100% of the female. So females either get red white or white white. There's no female with 100% red, and that's kind of what can happen. It could be other ones. It could be the Y ones. Only the male sons have it. Stuff like that, and it can really mess up inheritance patterns. But it's still relatively simple compared to some of the other stuff that goes on. Other ones is there's something called mitochondrial DNA. And that's DNA outside the nucleus of cell, and it's in the mitochondria. Now the mitochondria is usually called powerhouse of cell, but a lot of people makes energy for the cell. The mitochondria can have it does so many things for power, but it has linked to certain things causing deafness, blindness, cancer risk, decrease or increase, height risk, rapid aging, fertility issues. We don't know fully it's linked to certain other things, but we haven't studied that well. So it's really hard to tell how important it is, but it does exist. And it's much smaller than all the DNA in the nucleus. Usually, when the sperm goes into egg, none of the sperm's mitochondria passes on. So you just have an egg with 100% your mom's mitochondria. But for some reason, and we don't know how common it is, randomly sometimes that DNA survives, and you have DNA from both your father and your mitochondria. Some of your mitochondria is from your father, some of it's from your mom. And they don't mix up, they stay separate, but you have them both in you. And in cells, when they divide, they randomly split up the mitochondria. So you can have some cells with more of your father's mitochondrial DNA or your mother's mitochondrial DNA in that case, and so on and so on. And that can lead to more things because there can actually be an evolution of ways inside of your own body with your mom's mitochondria competing with your father's or so on. Also, one of your mitochondria, if your mom has an egg with some of the mitochondria mutated, that can happen too. So you can have one with more or less genetic diversity than a normal mitochondria, because normally almost all the mitochondria have very similar DNA, but you could have a case where half of them got mutated and have different DNA than the other half. So this leads to many complications. Another thing is that there's an environmental trigger or something might turn on or off genes, or choose what genes are on and off. So it's not, so the genes are on and off are environmentally dependent. And this is still one gene, may I remind you. This is not talking about many genes. And we can get to many, many genes. We're going to get to many, many genes. And sometimes more of one gene can actually turn that gene off. And this sounds counterintuitive because normally if you add more of something, it just increases instead of decreases. But this happens all the time, and we use it in genetic engineering for making plants of different colors, for new ways for fighting viruses, antiviral, for a bunch of things. And the simplest way it works, and there's ways that are much more complicated, is that how your body makes proteins, and DNA makes RNA, which makes proteins. And proteins are kind of what makes up most of your body and does most of the stuff in your body, is like chemistry-wise. RNA is usually single-stranded, unlike DNA, which has double strands, and its backbone is different, so it's less stable. And there's a couple other minor differences. But it's very similar to DNA overall. However, remember I said RNA is usually single-stranded. When RNA becomes double-stranded, it cannot make protein, and actually enzymes come out and cut it up. So what happens is, if you have too much of one type of RNA, that type of RNA can start interfering with itself, making more double-strand DNA that's cut up. Then you have amplification process, amplification stuff that goes on in your body that cuts up even more of that RNA until you don't have any more of that RNA to make the protein and turns the gene off. They just actually discovered when they're trying to make purple petunias by putting more purple genes in it. And instead they got a fully white petunia because what happened was the purple genes interfered with the purple genes in this exact mechanism. And then there's fancier versions of this mechanism that this is kind of the basis with, where you have more enzymes and other RNAs coming in and all this other stuff going on. But this is the simplest version of it. So that all flows away to inheritance, which means that it's hard to figure out if someone a gene is associated with, let's say, autism, or if a gene actually is causing autism in a person, because you have to look at how it's influencing each other with all these other aspects. You can't just say dominant or recessive when you look at two ones. But we have something now even more complicated, and that's because genes interact with other genes. Your height isn't usually determined by one gene. Your hair color isn't determined by one gene. Yes, there's genes that contribute a lot more, but there's usually not one gene. One or two genes determine if you're blonde, actually. But the exact shade of blonde, the exact how long it lasts, that's dependent on many factors. There's a few genes that we can determine eye color, but like the exact shades and stuff are still very dependent. But then height is really determined by many, many genes. And your brain is something really complex. So it's determined by many genes. And these can have stuff like feedback loops. And feedback loops are when one thing turns on one thing. In a positive way, a feedback loop is that it increases. So it's not just 1 + 1 = 2. Two things turn on thing. The two things interact and build up on top of each other. Like if you're in a car, and if a car accident, this is a horrible analogy, happens. An accident usually happens because if you were drunk, didn't have your seatbelt, and airbags weren't working, that's a lot higher risk for a bad accident than either one alone, and not just simply by adding them up together. A bunch of things are like that. But you also have negative feedback loops where stuff 1 + 1 doesn't equal 2, it equals something less than 2. It can even equal 0. And a negative feedback loop might be, you can think about taxes, that if you earn more money, that you might not fully start getting more money after a while because of taxes increase, but then there's really wealthy have ways around taxes and stuff. And there's also like being poor is more expensive. But like you might have other things that have negative feedback loops, like or don't increase at the same way, such as becoming a professional sports player. At some point, it's positive growth, but then you keep exercising and you don't get as much return for the same amount of exercise. You hit limits with a lot of things in life. So feedback loops can increase or decrease potencies of genes, and they can involve many genes interacting together. There's also switches in your body, and they may turn on or off other genes in certain ways. And some of these switches can even reverse the feedback loops once a certain state happens. So a gene very far away can turn on or off another gene. And then one way, or you might need two genes, and you might need mobile genes to turn on a gene. You might just need one gene to turn on a gene, but the genes can be located in different ones. So you can have a gene A or B in totally different locations on your chromosome to turn on a gene. You might need A and B. You might need something more complex to turn on a gene. You might have them interacting, like some turn it on, some turn it off. But you also have these switches, and these switches can go from negative to positive or positive negative, the same switch. Or might only be triggered once it hits a certain threshold. So this is how a switch could turn it to something positive to negative. Let's say more of a hormone builds up. And for a while, the switch isn't triggered until X amount of hormone hits. Then the switch triggers and a negative feedback loop happens and it turns off that hormone. Now everything is turned off and the switch can turn back on later, turn off later, and the hormone can build up again. And that can make a cycle. It can go up, down, up, down. And that's actually a big part of how all the hormone cycles, from the menstrual cycle to the circadian rhythm, work in your body. It's not the only thing that's going on. But you can have that with genes. And you can have all these other complicated switches that have, or you can have something that turns on after something different, X amount hits, that's totally different. Like, there's a ton of possible ones, and it's a whole network of hundreds of genes interacting. And also, like I said, genes can block or turn off genes that are very distant, sometimes on different chromosomes, totally different locations. So how does this kind of work? Well, a little bit is DNA makes RNA, which makes protein. Proteins either do most of the oversimplifying stuff. So RNAs can act as enzymes, but proteins do most of the enzymatic action, which means they do most of the chemical stuff in your body, like break down foods, digest them, make energy. DNA stores information, RNA acts as intermediate, and for like regulation of how proteins are made. Proteins also, the structural proteins that help make up like bone, nails, other things, along with other things like minerals and stuff like that. Hair has a high percentage of protein. However, RNA can affect, as we've previously seen with like the interfering RNA can affect how other RNA is turned into protein, both increase it or decrease it, or do something more complicated. Proteins can affect how RNA is made, how much RNA is made, can destroy RNA that is made, can modify how easy the RNA is turned into other proteins. And proteins interact with other proteins all the time. Proteins can help make other proteins. They can help stabilize proteins to keep them around. They can destroy other proteins. And all this different stuff. And then your DNA interacts with both proteins and RNAs. And your DNA has all the genes in your genome can be turned on or off to various degrees and stuff in between. DNA is a coil. It's a double helix. This helix can be wound more tightly, less tightly, and when it's wound less tightly, RNA can be more easily produced because stuff can fit in there, and it can make more. And RNA makes protein, so more protein can be made. Or DNA is also wrapped around a bunch of things in the body, it's wrapped around proteins called histones, and other things. And these ones can be wrapped more tightly, and less RNA can get into place, or more RNA can get in place and be made. DNA itself can have like chemical modifications or stuff not either directly attached or on the surface that can make it more easily or less easily read. Genes in your genome can actually jump around and move around your genome a little bit, and they can even replicate. This is how your body actually makes antibodies. It's with genes rearranging. Now, this doesn't happen a lot, but it does happen a tiny bit, and it happens in some systems super active. The systems that happens we know super active are antibodies, and we think maybe smell receptors. We think that different smell receptors are made this way. We're not 100% sure. But if you want to see something with active ones, look at corn with all the different colors. All the different colors in corn. The corn that's colorful is caused by genes jumping around. And it was how it was originally discovered. Now, you can also inherit different switches from people. So my switches might not be the same as your switches. You can handle different sensitivity to switches. You can handle different things to turn on and off the switches, the triggers. So you might have a trigger that's protein stuff that triggers switches more or less easily than me. And you can inherit everything you can inherit more or less, which makes this whole thing super complicated. And it's also why some people have act totally different, we know, in hormones. So some people, switches turn on more easily to make their bodies stop making as much hormones, so they don't have as much hormones. But at the same time, some people with less hormones of certain types, the body's more sensitive because the switches are more sensitive they inherited from ones, and certain switches in the body. In fact, we think the main difference in mammals and organisms is not now the proteins you make, but how much genes are turned on and off, and when they're turned on and off, and how they are turned on and off. What does this all mean for autism? Well, it means that it's just really hard to analyze what is causing autism, because the same gene depending on other genes or the amount or situation, it's still genetic, but it could be giving you autism or preventing autism from appearing in an individual. It could be doing the complete opposite things in different individuals, making it just so much more hard to analyze and figure out data, and you need better data sampling. It can also mean that autism, there's many different types of autistic people with different sensory sensitivities, with different ways of displaying it. And maybe the same gene gives rise to two totally different types of autistic traits in different people because of all this other genes and interactions. It means the data is just a lot harder to analyze. Now, if it's so hard to analyze, how have we done it? Well, we've done it because we've done it in really simple controlled systems, like flowers and corn that we've really bred well. And humans are not that simple system. They can really breed and do all these controlled experiments. And we're looking at one of the most complicated things, the brain, which we don't have a good animal model of for autism. And then we have it even harder that beyond all this, what makes it even harder is that we don't know we're going to see that complications even in reading the DNA. And I'm going to get to that in just a second. The same genes now also can be spliced in different ways, which is another complication, and this is not as an inheritance. And what this means is that one gene can produce different proteins. And I forgot to mention this, and one gene producing different proteins, the amount of each protein can be different in different individuals depending on other genes and environmental factors, which means I might have the same gene that makes an autism protein, so-called one, and a non-autism protein. But in me, it only makes the non-autism one, and you makes the autism one, depending on all these other factors, leading to even more complications, making this whole thing is hard to analyze. And autism is probably caused by many, many genes, and all the evidence is pointing to that. So then there comes the complications of figuring out, let's pretend Mendelian inheritance was fully right for a second. How would we find these, what is the problem then? Well, apparently many problems. One is that the closer a gene is to another gene, the more likely they're to be inherited together. And this can lead to false positives. And false positives are always a problem with small sample sizes in systems with many variables. The more variables, the more likely to find a false positive. There's more things that this might randomly appear things. But it's like, if you have a small sample too, take two people in a room, and you can find many false correlations. Those two people might randomly wear glasses for no good reason, might randomly wear shoes. You can find it all day long. But the reason why genes tend to group together is that you see that if they're close together on a genome, and this is a diagram showing it out, if they're close together, what happens is they're more likely to break them. Something's more likely to break apart away from them and bring them together when it crosses over. The breakers are more likely to go far away and bring them together. And if there's something that breaks them apart, there's most likely something that breaks them again back together. So you see less mixing of genes. We think that red hair and blue eyes might have actually been tightly correlated with the lactose tolerance gene and gene for light skin colors because they're all very close together. So red hair to not indicate light, none of these things should be correlated with the gene if you look at it a simple way, but they're all highly correlated because they're actually very, very close on the genome, unexpectedly so. But this can lead to a false positive very easily because you might be like, oh, this gene is interacting. And actually, sometimes it's easier to find these false positives than the other ones because oftentimes these false positives are things that are easy for the algorithm to detect because it's really hard to read the genome, and these false positives tend to be strong signals that are easy for the technology to read, like just single small changes compared to really complex mechanisms. And the false positives tend to have a lot simpler things going on than all these complex mechanisms where you have hundreds of genes interacting. Another huge problem, which is from the genetic basis of doing the study, is that DNA is not always the same length, even if it's in the same creature, and how the frame is moved is differently, and kind of also the grammar of it is different, where there is where we don't know where stuff stops and where genes stop or start very often, or genes can be mixed up in the genome, so they're not always in the exact same location from person to person. And so you can't just read it out and line up the genome. It's not that simple. For some people have longer versions of the same gene. Some people have shorter versions of the same gene. Sometimes people have stuff spaces in between. Sometimes people have multiple copies of the same gene in different ones. But to give an example in like English, I wrote down a couple sentences and you can come up with many more, but if I say, hi, my name is Bob, or I write down, my name is Bob, how do you line it up? Do you line it up by the first letter, or do you line it up how you best match it? Well, if you did not know the language English, you might not know how to do it. But I might have something that's even more complicated, like my, parentheses, ignore this, in parentheses, ignore, close by the name is Bob. How do you line that up? If you did not know what the statement is, you might not know to split up between my and name. So you might just line up by the my, the first letter, the first two letters, or you might line up at the end, or you might split it up by some algorithm. But we don't know those basic things, so it's really hard to line up DNA and figure out what actually is a gene many times. And we're still having that trouble, which then leads us to more biased results, because what we can figure out is a small sample, and it's more likely to have these false positives. Now, here's just some quick things about the study. 10K may seem like a lot of people, but it's still a small fraction of humanity, and especially if it's recruiting mainly Britain in certain areas, oftentimes it's going to be predominantly white, and that's going to even make the effective sample size smaller, because you're not sampling one, and they want families. Families are also more likely to give you these false positive correlations, because you more likely have families with genes that associate with each other to be done than an evenly mixed one from a broader category. You're also more likely to have not, because the other genes are similar, you might have less effects of the other genes that might also have an impact on how that one gene responds. If gene C is never present in your population with B, you don't know how these two things can interact. So get rid of these odd interactions I talk about, making results biased. The other problem with the sample and looking at genetic correlations with autism is that our sample is biased. This is caregivers and guardians and parents and stuff mostly signing up their kids that are distressed and think that distressed about their kids. So they're more likely to report very negative things about autism and see it as a bad thing. Or they're more likely to have kids with a lot of comorbidities they think are autism, but actually the comorbidities. And the people that are promoting it are people that have detested autism in the past and wanted to cure us and get rid of us. So again, lean to a biased sample. And this biased sample is not going to give us good data. Another problem is these people most of the autistic people in these studies tend to be non-consenting, and I went over that a ton in the Sparks video, which I recommend watching. But the reason why they're non-consenting is like there's consent forms and everything, and they'll talk about consent, but these are the parents and caregivers, and there's instructions for Spark at least of how to get DNA from unwilling young kids, such as giving them food that makes them salivate, taking samples while they're asleep, while they're stooling, and using chew toys, and even forcing them in the hospital to give them medications to make them spit. This is all on the website, and most of Spark's participants were non-consenting young kids. So that way biases your sample for one thing, because that tells you something that it's not ethical. These people want results very much, so that's a huge problem. And they want money and this one. And also another problem is like You might talk about like, oh, it's important to study autism because of all these comorbidities. Why don't we study the comorbidities? Instead, you can have a better sample size without, if you want to study, for example, okay, I'm going to give a specific, Ehlers-Danlos syndrome or gastrointestinal problems, and I might be pronouncing some of these wrongs, or other things that are more common in autistic people. They also occur in non-autistic people. And if we look at the wider, that bigger sample, we're like less likely to get false positive with this autism stuff, and we're more likely to figure out a better treatment that doesn't hurt autistic people, change autistic people, but helps a wider group of people. Also, this data is not regulated. Oftentimes how they analyze it, and they don't have to tell you how they analyze it, and that's a huge problem. The FDA does not approve genetic testing in the US almost at all. The same as in the UK, Australia, and Canada, and most places. Meaning your test doesn't have to go past various rigorous standards. It can be complete BS. And oftentimes, a lot of genetic testing is complete BS. There are good genetic tests, but those are for a few genes, like BRCA1 and 2, which really can dramatically increase your chance of breast cancer. But those tests are actually patented because they're so good. And that's also really messed up that they can be patented. But when you're talking about like how are you likely to be bald, what age you're likely to die, all those are not regulated, and they can give you very consistent results depending on who you get it from. And also, they're allowed to sell your data elsewhere, which is another huge problem. And really will give up privacy and monitoring. And they're more likely also, if they have this data on an autistic individual, they're more likely to stalk that autistic individual, get more data, because they want correlations with one individual to follow one individual around, instead of trying to find another individual and also sequencing their DNA. But all of this, because it's so complicated, we're oversimplifying it and medicalizing it, it just further stigmatizes autism, leading to further medication. And this stuff is really complicated, and the unknown is scary, so it makes it sound even scarier when you start saying really complicated things like FOXP2, multiple Mendelian inheritance, and people get scared and start believing in fake nonsense because people start peddling cures of like that sound scientific, but they're just words that mean nothing. Now, what about leading this to a drug for autism? Well, if we're basing a drug just on genes, we can make genes that turn on and off different things related to that gene. But first of all, is that gene truly causing autism or not, and how it is, and so on? I've always shown it's super complicated and we don't understand it. And like inheritance is super complicated, so maybe turning on off the gene is not relevant. It's a complex multiple inheritance involving hundreds of genes. So the drug can have a lot of side effects, and it contains a person other things. So we don't know how all these genes interact. We have to look at the whole, not the one single little gene like the cortisol in this complex system. Who decides gets the treatment for a young kid if they're drugs? Or the panicking adults that do not give the child alternatives, ways of teaching the child when the child is to fit in, force child to speak, put them through therapies, sometimes over 80 hours of therapy a week that are abusive, so to cause PTSD, or the ones that let them live and give them alternative means and access. And we promote more and more fear and medicalization. Instead of learning to adapt to them, we just give them drugs. And also these drugs don't target the one gene or stuff well, or protein in the mix, which I've always said might not be the way of looking at it, because you have to look at the whole system as a whole. But they oftentimes have off-target ones, and we're not good at making off-target ones, which further makes them more side effects. And beyond all this, the money is better spent elsewhere. The other alternative that these genetic testings could lead is to not having babies, or having that's autistic, making us go extinct. Or it means that you have stigma as a child and only placed in harmful therapies as a child. And with these tests, we don't know the reliability of it. It may lead to false positives and negatives often, which can have both harms and cause harms in both cases. And beyond causing false positives and negatives, you might have people are more likely to see other correlations. I've seen this in Spark for Autism one, where parents will complain that the child had the worst form of autism for 2 genes, but something appears in 25% of the Caucasian population. So like, clearly something is off, but they don't understand it. So they might look at this and think that this gene causes this type of autism, but that's a false correlation for all the reasons I'm saying. And the child doesn't have light sensitivity, but sound sensitivity. And instead you're doing sound sensitivity and just looking for light sensitivity you don't have. Or you make up all these assumptions about your child based on genes that are not true, because the system is so much more complex. Later on, I'm going to make some follow-ups to get into this one. But also the brain is way, way too complex to simply understand from genetics. So we're not going to understand how the autistic brain thinks from this. This epigenetics, which is the cool thing about how between environmental and genetic inheritance, how this is wrong from an evolutionary standpoint. Briefly, that we live in a society, so it's not about being fit always. It's sometimes being fit. Sometimes society adapting to us makes the society stronger as a whole, such as an individual honeybee or a beaver cannot survive well, but beavers build their own homes to survive well. So, but they have to build their homes. Honeybees, the male bees won't survive at all, and the drones and worker bees, but they live in a society that they all work together. So you have to look at how society functions for evolution, and we're horrible judges. And it's just also, again, it takes money away from more valuable things. And it's going to be millions to billions of dollars spent on this. Well, thank you very much for listening. I hope you liked this and found this interesting. Please like and subscribe. It really helps the channel out. I'm trying to reach 1,000 subscribers in a year and 4,000 watch hours, which is really hard, but really help. I think I can do it. Thank you very much. Goodbye.